to our next session, which is our Mini TED Talks session. We got a lot of great feedback on this last year, so we've brought it back and hope it's as fun as it was last year. Chairing this session, please welcome Travis Besanger, Senior Business and Technical Director for Kinectrics.
Okay, good afternoon, everybody. I have the honor to introduce our speakers today for the Mini TED Talk session. And this is a session really looking on what's next. For those that have been in the industry for as many decades as I have, we've really seen a change in the industry, really a resurgence of the nuclear medicine industry in the last, I'd say, 5 to 8 years. That began with Xofigo, sometimes forgotten drug, and actually the first alpha therapeutic to hit the market, and has really grown with the introduction of Lutathera and Pluvicto. So we've seen huge investment come into the industry, massive innovation. It's really the most exciting time in nuclear medicine in decades. So today, we want to see what's going to be building on this foundation and looking beyond the agents, both therapeutics and imaging agents on the market, and seeing what's next. So with that, the speakers that we'll introduce today will be looking at what is the next step in both oncology and non-oncology indications. So our first speaker is Dr. Kim Allman. Kim Allman brings over 20 years of oncology expertise, focusing on GU cancers, particularly in renal cell carcinoma. She's the medical director of Radiotheranostics at Telix Pharmaceuticals, and she leads the efforts to develop novel targeted radiopharmaceuticals, both diagnostics and therapies. These are going to be rapid sessions, so I will leave the time to our esteemed speakers. So Kim, welcome to stage.
All right, well, thank you for that kind introduction. And I will emphasize that my background is in medical oncology. And the reason that I do that is for 2 reasons. First, to tell you that I am really humbled to come into this space. You know, I've worked with targeted therapies, small molecules, immune therapies, chemotherapies. Radiotherapy is its own beast, and I am humbled by all of the intelligence in this space that is really advancing oncology along. But secondly, I'm telling you that with my background to really relay firsthand the impact that this is going to make on patients and has already in the prostate cancer space. For those of you who don't have a scientific knowledge or background about how prostate cancer is treated, this is really kind of our model for radiotherapy. So I just want to walk you through that just briefly. So the foundation of prostate cancer is to cut off testosterone production, which is done through chemical or surgical castration. And then really with each type of cancer, we move into different eras of treatment. So one of the original eras was chemotherapy, which is very common for all cancers. But it's extremely non-specific. It's kind of targeting all cells, leads to a lot of toxicity. And so then within prostate cancer, we start to move in a different direction and be a little bit more mindful of what is the biological driver. Again, it's testosterone. So perhaps we need to focus on developing therapies that block those pathways, and thus those were created. So really, those are the major kind of eras that we've walked through, and then we come into the biomarker era, right? So what does prostate cancer express uniquely so we could be even more precise? Well, it's PSMA. And so that's how different therapies, both diagnostic and therapeutic, have come along. So if we can do that in prostate cancer, can we not do it in other cancers? Can we do it in kidney cancer? Can we do it in brain tumors? Similar to what Dr. Dancy had mentioned this morning, can we do it in pan-cancers across the spectrum? Is there some biomarker, receptor, target, transmembrane protein that we can exploit for our benefit? And that's really what Telix is trying to do. So we have a company motto of see it and treat it. So we're looking for both the diagnostic and therapeutic capacity to treat all types of cancers and cancer ubiquitously. And if we have the ability to target all types of cancers, can we be agnostic about our isotopes? We use gallium, we use lutetium, we use alphas, we use zirconium, iodine, you name it, whatever the situation calls for. And then can we change our vectors too? What does the disease require the most that gives us the best precision or the best targeting and minimizes off-target toxicities? So I'm going to open up with my personal attached molecule. So this is girentuximab, which targets CA9. So just to indulge me a little bit in the science nerd in me, I'll just explain why CA9 is relevant. So we know tumor suppressor genes are kind of our body's inherent functionality to stop things that are growing aggressively because they probably shouldn't be there. So if we lose functionality of that, there's a series of cascading signals that set off other things, ultimately which can lead to upregulation of CA9 in clear cell kidney cancer. So that's why this is particularly relevant to me. So girentuximab, this monoclonal antibody, specific and selectively targets CA9, which is ubiquitously expressed in clear cell RCC to the tune of about 95%. So you see here, with that same underlying vector, we have the ability to both diagnostically assess with zirconium and then also therapeutically treat with lutetium. Not represented on this slide is our alpha program, which is kind of the next iteration and earlier in development. But I will share with you that the zirconium girentuximab is about, has a PDUFA date in the US of about 10 weeks from now. So as I mentioned, we've gone through these different areas in the therapeutic side of oncology, but also in the diagnostic side. So as it pertains to kidney cancer, we really have limited options in terms of our imaging modalities. We have CAT scans and MRIs, which tell us a great detail about structure and vascularity, but they lack the ability to distinguish between benign and malignant lesions. So right now, in current practice, urologists are really basing these decisions off of imperfect criteria. What is the size of the tumor? How fast is it growing? So by going off of those metrics without an understanding of histology, you could make an inaccurate decision where you could do surgery in a benign condition or not intervene in a malignant condition, which really tells us that we could have some enhanced capability if we use something that is uniquely expressed to the cancer. So FDG is a modality that is available for RCC, but it's imperfect. You can see it's renally excreted, so there's a lot of obstruction to the images. But when you can see with zirconium girentuximab, it is specifically located just to the renal lesion alone, that indeed is malignant, and thus the patient and clinician can make a treatment decision together. So if we can do it in just the kidney itself, can we do it in the rest of the body? Yes, we do know that these biomarkers are expressed in metastatic sites as well. So we have the ability to leverage the same kind of original capability to look at whole body assessment. So here in this patient, we can see kidney cancer is expressed not only in the primary kidney, but lymph nodes, lung, soft tissue, and also other areas within the body. So the ability to detect it within other organ systems. And so if we can do it in the kidney, can we do it across tumors? So CA9 is actually expressed in about 13 different tumor types demonstrated preclinically. So perhaps we can, and we're starting to explore that. So in this first rotating MIP, this is a patient who has clear cell RCC, and as I expressed, we know very clearly expresses CA9. So if a patient has a solitary renal mass, this patient would generally go to surgery if you knew that it was large enough and cancerous, and they could be in a curative state. Well, I don't know if you can see from this rotating image, but when it flips around the back, you can see the lesion on the upper part of the posterior rib as well, which means that the treatment plan completely changes for this patient. Perhaps we do a metastasectomy where we remove that, or do SBRT, or the clinician may feel that it's appropriate to start systemic therapy. Regardless, not only does this give us accuracy in imaging, but it also can change clinical management as well. So in the next graphic, you can see the CA9 targeting radio tracer looking at a patient with mesothelioma, which also expressed CA9. So you can see how we're starting to move into this more pan-cancer state, and then similarly to colorectal as well. That last trial that's represented is a therapeutic trial that we actually have ongoing in Australia, in which it's a pan-tumor population where they're imaged with the zirconium girentuximab followed by dosing of therapeutic lutetium girentuximab. So it is a see-it-treat-it situation. And one more target to mention, if we can do it with CA9, are there other proteins that we can exploit similarly that are diffusely noticed throughout the body? Fibroblast activating protein, or FAP, is one of those two as well. And so that plays a really important role in the tumor microenvironment by putting this protective layer on the extracellular matrix. And it is expressed in all of the tumor types you see here that have a purple dot next to it, where CA9 is represented with the blue dot. So you may have heard about FAP previously, and there wasn't a lot of great residency or internalization times. So Telix has acquired a molecule that has kind of this dual-pronged approach where it can start to target with two arms, so it can pull itself in and stay within the tumor, and then has the therapeutic payload on top. And last, I'll leave you with the image, because talking about science is great, but applying it to patients where this really makes a difference is where this does make the difference. So this is a case of a patient who had diffusely metastatic breast cancer, who was imaged with FDG initially off to the far left-hand side, and then FAPI PET immediately next to. So you can see that the patient received then subsequently several cycles of therapy with a lutetium-based FAPI. So then already at the interim assessment, you can see that that disease has really started to reduce. With subsequent cycles, that's what leads to that interim assessment on the far side, where the disease has really melted away. And you can take a look at the SUV has dropped by about tenfold, which is really a huge win for this patient. So I'm over time, but I thank you for your attention.
Okay, next up we have Dr. Carlos Uribe. Dr. Carlos Uribe is a certified nuclear medicine physicist at BC Cancer, specializing in dosimetry of radiopharmaceutical therapies. Co-lead of the Current Laboratory, he advances quantitative imaging and personalized cancer treatments. Carlos also gave a wonderful talk last year, so I know we're all going to be in for a treat. His talk today is Seeing is Believing, Why Post-Treatment Imaging Matters in Radiopharmaceutical Therapies.
Okay, I think we were not going to cry today, but we all know this patient, and it's a man that was injected with some lutetium therapy for his prostate cancer, and his PSA is going down, which is pretty awesome, but we have no idea where the radiopharmaceutical went, right? And I don't know if you guys are comfortable not really knowing where the radiopharmaceutical went. We're hopeful, but we are blind. And we keep talking about precision medicine is all around the place. However, in theranostics, when we talk about this, we know the pre-therapy PET. So we kind of know where we're starting from. We see the target there. But when we go to post-therapy, it's like opening a book where this page is intentionally left blank, and then we go and inject again, and it's like the next page is also intentionally left blank. So I don't know. We don't really know if the radiopharmaceutical is going to the size that we think it is going, or if it's missing some. So I want to tell you guys what is that we're missing if we don't look. And this is a first example. What you see at the left is a PET image for like the diagnostic PET for this patient. And this patient was accepted to therapy. Therapy started 2.5 months later, and there was a SPECT done. So you see there's a difference between that diagnostic PET. We're not really starting from where we think we're starting. There's some new lesions that are showing up. This is a patient that has been treated for neuroendocrine tumors with lutetium dotatate. We have a baseline PET, then the post-treatment SPECT images show that the lesions are that they're responding. So for example, in this case, this patient, like the physicians, decided to stop treatment a little bit after cycle 3 just to save some of these cycles for when that disease came back because the patient was doing very well. This is the typical, well, not the typical, but look at this patient. We have a patient that we did some labs. Cycle 1, PSA 60. Cycle 2, PSA 12. Cycle 3, PSA 7.6. Oh my God, this is amazing. PSA is going down. This is what we all want. And we bring the SPECT. Well, look at the liver. So look at the arm between cycle 1 and cycle 2. There's a lot of new metastases coming out in the bone lesions of the arm. The liver seems to be responding on cycle 2, but then the disease progresses again in the liver on cycle 3. If we just take to the labs, we would think this patient was doing amazing. There's some other things that we can note from this SPECT imaging, SPECT-CT imaging. We see new PSMA avid lesions like the one that I'm pointing with the arrow on the leg. But just by looking at the CT of the SPECT-CT, it's not even a diagnostic CT, we see that there's a new lesion that is not PSMA avid in the liver. So my point is, post-treatment SPECT is not just it's just basically adding us information that we don't really see in the labs or in just convention or in other conventional imaging. Now, does it have real-world impact? Well, it turns out it does have real-world impact. It's not just something that I'm saying. There's been 2 studies, one for neuroendocrine tumors on the left, one for prostate cancer on the right. Patients that got post-treatment SPECT-CT, there was a change in management in 27% of the patients for neuroendocrine tumors and in 50% of the patients for prostate cancer. What type of changes? Well, maybe some injections were delayed, maybe some injections were interrupted. You can go and look at those articles. The tables are there clearly, but that's the type of management. So this morning we heard, I think it was Stella that was mentioning, well, about to do these kind of things, we need some guidelines, some procedure standards, and that's exactly what I did. I gathered a group of experts around North America that involves a lot of nuclear medicine physicians, a lot of physicists, and we started putting out some procedure standards that went through some public review and are almost coming out for the public publication, official publication at the end. But what we're saying is every patient treated with lutetium therapy should have a post-treatment image, a post-treatment SPECT-CT. Now for prostate cancer, it should be after every cycle. For neuroendocrine tumors, after looking at the evidence, we saw that maybe for higher grade, like grade 3 tumors, it should be after every cycle, but maybe grade 1, it can be every other cycle. And for grade 2, we don't really have a full consensus just yet. Now, this imaging should be done in a quantitative SPECT-CT, and what that entails is calibrating the equipment, following some protocols, because it's going to provide us some information that also several studies have shown that SUVs and some of these optics starts correlating with better prognosis, and we can predict some of this. We discussed a lot about having multiple SPECT-CT scans or just a single point imaging scan, and well, we think that in a clinical practice, we need to make it easy. We need to start from somewhere. A single point is acceptable. We know that if we want to then move forward to performing dosimetry, well, maybe we want to have more than one scan, but we already have some methods that can give us a good approximation with just a single scan. When do you do that single scan, though? And then we debated that a lot. And, well, we found out that maybe imaging the next day, around 24 hours, is very, very good for qualitative assessment. Maybe later is preferred for dosimetry. If you want to do dosimetry for prostate, you benefit from a scan around 48 hours. If it's for neuroendocrine tumors, you benefit from a scan about 72 hours for the purpose of dosimetry. However, we also discuss even imaging the patient 4 hours after injection, because we understand that some people, and we saw some examples this morning, once they're in the hospital and they leave, it's hard to bring them back. And there's still some benefit of doing a 4-hour imaging that maybe for qualitative purposes can compare to the 24-hour. It's not the best for quantitative dosimetry, but it's a good start. Well, we need to ensure that those scans have an adequate anatomical coverage. We want to be able to see the organs at risk. We want to be able to see all the lesions. So it's typically from top of the head to mid-pelvis. And with a standard SPECT scan, it depends on the height of the patient. It's typically 2 to 3 beds. And lastly, what I want to add is you can consider this as an alternative to interim PET-CT. Now here, we have already injected the patient, so in a way, the radiopharmaceutical is free. We don't need another radiopharmaceutical to do the PET. And so a lot of these SPECT images provide very similar information to what you would observe on a PET, so it's something to consider. I think this is the first step towards personalization. If we start doing this imaging and we start using this imaging for that response assessment as part of the therapy, then there will be a natural transition into personalization, and we start getting more familiar with how to incorporate this into the clinical workflow and how to start doing our SPECT CTs. And I just want to close saying, if we're not imaging after treatment, we're guessing. And I guess I showed you guys multiple examples. Thank you very much.
Okay, up next is Dr. Amanda Boyle. Amanda Boyle is an assistant professor at the University of Toronto and a scientist at the Centre for Addiction and Mental Health. She has 15 years of experience in PET imaging and radiopharmaceutical development. Her research identifies biomarkers for neuroinflammatory and neuro-oncology conditions, applying radiotheranostics to bridge oncology and neurology. Today, her talk will be Beyond Cancer to Inflammation, Expanding Canada's Imaging Capabilities. Welcome, Amanda.
Thank you very much. Positron emission tomography is a powerful molecular imaging tool that can be used to visualize biological processes in the human body. Arguably, oncology is the most common area of study that's studied with PET imaging. Other areas include cardiology and neurology. All of these areas have disorders that involve inflammation. Today I'm going to talk about how we can take advantage of this common ground seen in inflammation and different disorders to expand our capacity for PET imaging. Oops, did that go too far? I don't know. In the periphery, we have the immune and inflammatory cells, macrophages, T cells, and B cells. The blood-brain barrier prevents these cells under healthy conditions from entering the CNS. In the CNS, they have their own inflammatory cells. These are microglia, oligodendrocytes, and astrocytes. Any of these cells are valid targets of PET imaging of inflammation in many different disorders. In a disorder like the autoimmune disease multiple sclerosis, this leads with a leaky blood-brain barrier in which the peripheral inflammation cells, macrophages, T cells, and B cells can enter the CNS. So sometimes you'll see all of these cells in one place. So those are the cell targets that we can try to image with PET imaging, but we can also take advantage of the dysregulation of inflammatory pathways in non-inflammatory or immune cells, such as malignant epithelial cells. Some of the pathways that you would have heard of most likely that can be dysregulated are MAPK, TNF-alpha, JAK-STAT, and prostaglandin synthesis pathways. I'm going to use the prostaglandin synthesis pathway as an example and show you how we can PET image COX-1, cyclooxygenase 1, in cancer, move beyond cancer, and then image multiple sclerosis with this same PET radiotracer. There's high COX-1 expression found in high-grade serous ovarian cancer, which accounts for 75% of all epithelial ovarian cancers. So preclinically, we looked at PS13, which is a highly potent, highly selective COX-1 PET radiotracer. We looked at it in ovarian cancer tumor xenograft mouse models. And indicated by the white dashed circle, we were able to image these tumors very nicely, and we could displace the PET signal very nicely with the COX-1 inhibitor ketoprofen. So then we move on to looking into this in a mouse model of multiple sclerosis. So as mentioned, there's the leaky blood-brain barrier. So where is COX-1 going to be found in this model? Well, macrophages and B cells will be there, the leaky blood-brain barrier. Those both express COX-1, as well as microglia, which is apparently, this is where COX-1 is primarily located in the brain. So we used PS13, and we looked at this in a mouse model of multiple sclerosis called experimental autoimmune encephalomyelitis mice. These mice gain clinical symptoms that increase in severity from 0 to 5 as the scale, and we used mice with a scale with an EAE score of 3. That means that their hind legs were paralyzed. And in these mice, indicated by the red arrow, we saw an increase in PS13 uptake in the cerebellum. And this was confirmed with our volume of distribution quantification. So the healthy controls shown in blue, the EAE mice are shown in red. So this is a really nice result for this study because the cerebellum is responsible for balance, posture, and muscle coordination, which problems with gait and muscle coordination are the most common symptoms of multiple sclerosis. So this is a very promising tracer for multiple sclerosis. So you may look at this COX-1 story of imaging cancer and then imaging multiple sclerosis and say, cool, so we can repurpose a PET inflammation a PET tracer that targets inflammation. But where's the therapy aspect for the scope of this radiotheranostics program? Well, at CAMH, we use small molecules. We only radiolabel with carbon-11 and F18. A lot of our small molecules are actually kinase inhibitors. So these, when unlabeled and given at a higher dose than what we use with PET imaging, have the potential to be used therapeutically. I'm going to do another example where we tried imaging multiple sclerosis mouse models. So Bruton's tyrosine kinase, or BTK, is an intracellular molecule found in B cells, macrophages, and microglia. Ibrutinib is a BTK inhibitor that's been used for many years as a chemotherapeutic drug for B cell malignancies, such as lymphoma. And we radiolabeled ibrutinib and put it into our EAE mouse models. We found that ibrutinib did not enter the brain, though we did see an increased uptake in spinal cord, which is promising. I do think that if we had a BTK tracer that more readily entered the brain, that we would have a more promising a PET radioligand, but it's not great in this example. Still, in this paper that I found from 2023, they used a therapeutic dose 10,000 times higher than what we use with PET imaging, and they were able to reduce the severity of clinical symptoms in EAE mice. In this graph, you see in blue is the vehicle, and then when they deliver a therapeutic dose of ibrutinib, 9 days, beginning at 9 days post-EAE immunization, they have a reduction in clinical symptoms. This is further reduced when they begin the therapy at 0 days. And so in summary, repurposing PET radioligands in different diseases allows the identification of new diagnostic strategies for different diseases. And visualizing the distribution of a radiolabeled drug in vivo can speed up drug discovery. Maybe we want to look for new chemotherapeutic agents. And my favorite thing about doing this kind of work is that it opens up interdisciplinary collaborations. So we can move beyond cancer, and if anyone knows anyone who has any interesting preclinical inflammation models, please let me know. Thank you.
Okay, last up, we have Dr. Madhura Naik. Dr. Naik is a physician researcher at Novartis. She leads Canada's radioligand therapy portfolio. She has over a decade of oncology experience and has led the pre-launch of Pluvicto. She's also initiated over 11 global clinical trials and applies real-world evidence in projects to advance radiotheranostics for solid tumors. So welcome to the stage, Dr. Naik.
Thanks for a very kind introduction, and good afternoon, everyone. Those were some tough presentations to follow. It's truly a matter of honor to be present here in front of this audience, and zero pressure presenting to a room full of experts on radiotheranostics about radiotheranostics, and representing the work that we at Novartis are doing globally in a span of 8 minutes. But I'm going to try my best. I want to start off by talking a little bit about radiotheranostics and where we've come from, because it's important to know this before we know where we are going to go with this therapeutic. And I think I want to talk about radiotheranostics, and I think Dr. Kumna also mentioned this in her panel discussion. This is not a treatment that's new. This has been around for decades. And really the first foray into radiotheranostics began with radioactive iodine, as we all know. But radium-223 was the first therapeutic modality to show clinical application in prostate cancer. From there, the first radioligand therapy that got approved and got access for patients with neuroendocrine tumors was lutetium-based oxodoturitide, or Lutathera as we call it. But if we talk about the real inflection point in radioligand therapies or radiotheranostics, it was the introduction of PSMA-617 in the treatment of prostate cancer. And if you allow me, I'd like to share a personal anecdote about this. And we were, as a team at Novartis, looking at the first data that came through from the phase 3 clinical trial with PSMA radioligand therapy in prostate cancer. And we thought to ourselves, this is not just data that's statistically significant, it's potentially paradigm shifting. Because a treatment modality that we had thought of as restricted to niche types of tumors or had only read about in academic papers was now slowly going to become mainstream cancer pillar in the treatment of cancer. And this was both exciting and nerve-wracking. And building on our success with PSMA radioligand therapy and SSTR-targeted therapies, we've learned a lot along the way. But I'm not here to talk about that. I truly am excited to share what radiotheranostics can now do in the future. And when we look at Novartis, we think about ourselves as a company of firsts, because we've been the first to launch commercial radioligand therapies for neuroendocrine tumors as well as prostate cancer. But we are truly also the first in the industry to have the breadth and depth of portfolio in radiotheranostics that's trying to tackle the most important challenges that are facing cancer treatment today. And the way I want to talk about expanding the blueprint from Novartis is by discussing a few key approaches. And I've really tried to consolidate a lot of the work that we are doing from preclinical research onto late-phase clinical trials in this one slide. And if you allow me, I'm going to nerd out a little bit. But the first approach that we are taking as a company is to expand the use of radioisotopes beyond lutetium. And we've heard a little bit about actinium-based PSMA radioligand therapies, and we have two key assets currently in clinical development in phase 2 and phase 3 studies with actinium PSMA-617 and actinium PSMA-R2. Not only are we evaluating these assets in late metastatic castration-resistant prostate cancer, but we're also evaluating them earlier in metastatic hormone-sensitive prostate cancer. With our advent and entry into actinium-based radiopharmaceuticals, we're now trying to target PSMA-based radioligand therapies that can potentially have greater potency, efficacy, and lead to better clinical outcomes for our patients. The second key approach, and I think we heard a lot about this in Dr. Janet Dancy's talk today, that PSMA and SSTR are truly just the beginning, and we have a lot more different molecular targets to look at. And Novartis is also really expanding the ways in which we look at these targets. I want to mention a few that are of interest. And we just heard about the FAP theranostics that Telix is looking into. And I'd like to build a little bit more on that. The first area of research that we're actively looking at is actually FAP, or fibrinogen activating protein, fibroblast activating protein. This is an area of research that's very interesting because we're not only looking at tumors, we're looking at targeting tumor microenvironments. And this can have potential ramifications across indications in oncology. We currently have basket clinical trials running for FAPI theranostics, and we're specifically looking at some key unmet needs in cancer, namely pancreatic cancer, lung cancer, and colorectal cancers. The second important target I'll speak about, which is very unique and interesting to me, is GRPR, which is gastrin-releasing peptide receptor. In early clinical research, we found that this is a signal that's overexpressed in breast cancer, and we are now evaluating this specifically in phase 2 and 3 clinical trials for patients with advanced breast cancer that have progressed on CDK4/6 inhibitor therapies. The third target is DLL3, or delta-like ligand 3. Again, for those of you who work in this field, you're aware that this is a target that's associated with aggressiveness in cancers, especially small cell lung cancer. And this is an area that we are actively pursuing. The fourth one that I'll talk about is HER2, And HER2 is actually an important target, as we know, for breast cancer and lung cancer. And HER2-directed radioligand therapies are an area of research that we're looking at actively, with first-in-human dosing studies already underway. Now, when we look at these targets, we're not only looking at tumors that are high incidence and prevalence across the world, we're looking at tumors that have the highest unmet need. And oncology's most challenging indications are now going to become RLT's new focus. So we're talking about triple-negative breast cancer, we're thinking about pancreatic cancers, we're talking about small cell lung cancers, and some rare neoplasms, including gastric cancers. Now, it's no specific cancer therapy pillar can stand on its own. And therefore, lastly, I want to talk about combinatorial approaches, because we would like to fully leverage the expertise and the therapeutic efficacy of radiotheranostics by looking at combinations with other treatment agents. And we're looking at 4 different approaches here. The first one, to combine radiotheranostics with chemotherapy, to look at combinations of radiotheranostics with immunotherapies, but also combine RLT agents with therapies that can target newer targets and newer biomarkers. EZH1/2 inhibitors are non-RLT assets, but are unique targets in the form of gene-directed therapies. And these combinations can potentially have better clinical outcomes for patients with prostate cancer. The second important target is, or a type of therapy, is actually novel AR degraders. And these are therapies that can have potential to treat prostate cancer in patients that have resistance to ARPIs or conventionally available therapies. Now, when we look at these combinations, we are moving beyond prostate cancer to also evaluate these in treatment of breast cancer. So truly what I want to emphasize on is that these clinical programs or these ideas are not just concepts on paper. These are actually clinical trials that are ongoing as we speak in the form of IND-enabling studies, phase 1 and 2 expanded cohorts, as well as first-in-human dosing studies. And as we move forward, we need to realize that for us to talk about the promise of radiotheranostics, it's not enough to just talk about clinical development or clinical trials. Because radiotheranostics is not just a form of drug or molecule, it's truly an ecosystem. In the pharmaceutical industry, we like to talk about launch readiness or launch engines, but with radiotheranostics, we need a very different kind of readiness, a different kind of engine. This engine is driven by various different parts that need to work together in close synchrony for us to achieve that final goal of getting these therapies to patients. Not only do we need the oncology expertise, but we also need the nuclear medicine infrastructure and training. We need focused medical education for professionals and healthcare providers that have never touched a radiopharmaceutical before. We need expertise in diagnostic imaging to make sure that we have the right pairing of the diagnostic agent with the therapeutic agent. We also now we are thinking about supply chain, something that we've never talked about for any other oncology agent before. We need to think about adaptive dosing and novel clinical trial designs. And in all of this, we're also thinking about the new wave of AI and how we embed artificial intelligence and deep learning into all of these different parts of the engine. Importantly, we also need to involve and talk about policy making and reimbursement, something that you will hear more about in the panel discussion at 4:25 p.m. And our VP from Novartis will also be present and talking about how Novartis has led the way to bring these therapies to patients, not just in Canada, but across the world. And so what's important to note here is that we require a very different kind of mindset as we move forward into radiotheranostics. And if I want to talk about the next 5 years or the next decade, we are truly going to see a real shift. And we are going to solve not just the problems that I talked about before, but even more increasingly complex challenges, such as figuring out dosimetry, understanding how to deal with short-acting radioactive isotopes, figuring out how we should manage toxicity and adverse events of these agents, and also handling variable tumor expression of many of these biomarkers that I spoke about. In this future, we are going to be dealing with a lot of chaos and confusion, and this can truly cloud our conviction in radiotheranostics. When that happens, I would love for us to go back to this image that you see on the screen. This is an image that all of you may be very familiar with. Some of you probably have this tattooed on your bodies, if not on your brains already. This is the SNMMI image of the year from 2018 from Michael Hoffman and group, which showed the 8 patients that responded to PSMA radioligand therapy with extremely good clinical responses. Something that we had thought was out of our reach, was extraordinary in the field of cancer treatment, has now become the benchmark for treatment in cancer. This is what we aim for with the targets and the unmet needs in cancer treatments that I spoke about. And where I stand as not a physician at the bedside of a patient, but someone who's helping translate clinical science into strategy, I don't see radiotheranostics as just a promise, because a promise sounds passive. I see radiotheranostics as something that's inevitable. Thank you so much for your attention.